Okay, so this question looks relatively difficult, but I'm going to explain it and you will see that take 2 minutes, 30 seconds. So I will explain it very slowly. So later, if you have it in a test, you can answer it very fast. So the first thing is that they are giving you an example of Drosophila, and they have mutations, one in the eye color, and the name of the mutation is scarlet, and I will talk about that. And there is another mutation that is in the bristles, modified the bristles, and the mutation is spineless. And these are the symbols. And here is the most important part of this information. This is not very important, the name of this mutation or this one, we can change it. However, the most important part is this, that they are in the same chromosome, located in one chromosome, this case the third one, and that the distance is 14 MAPS units. That is the most important information to answer this question. And I will show you in a second why. And they want to tell you, they want to ask you what phenotypes and in what proportion would you expect the progeny from the mating of these two genotypes for this genotype for females and this genotype for males. Okay, so they're telling you that these two genes have a distance that we know. So when you make this cross, what do you expect? What will be your expectations of the genotypes and the progeny? Okay, so it's pretty easy. Let me show you the answer. So the answer is very easy. So the parentals, so you see here, these two will have 46%, one will have 43, and the other one will also have 43, while the recombinants will be 14%, because it's the distance that they give you. Each one will be 7%. And how do you come with that answer? So pretty easy. Let me show you in a second. So first thing that I want that you remember is that this very easy diagram that they show you in your textbook, there is a section A and there is a section B. In the section A, they show you an example when there is no crossover. Yeah, so you have one chromosome that is blue, that is the parental, and one chromosome that is green, that is the parental. And here you have wild type plus is represent is the symbol for wild type, and here they have possibly two mutations, but they are shown here. If you see, as there was no recombination, the outcome is that there is four recombinants, four non-recombinant chromatids, as you can see here. So you get exactly the same combinations that you saw in the parents. Yeah, so blues with A and B, and greens that are wild type. Good. So 100% of them will look exactly like the parentals. Yeah. However, in the schematic B, something different happened. Here you have crossover. So as you can see, the green and the blue are mixing. So here you see the chiasmata, and as a result, you will find here pieces of the chromosome that are combining. Yeah. So what you're going to have here is a non-recombinant, that it will be this one, the entire blue or the entire green, non-recombinant, and you're going to have recombinants that are the ones that are in between. Okay, so in conclusion, you will have here a different proportion. You will have here, this will not be, the parentals will be lower than 100%, yeah, and the recombinants will be higher than 0. Yeah, well, here in the non-crossover, the recombinants were 0. Here the recombinants will actually appear. Okay, so let's see with our examples what is happening there. So one thing that I told you is that you don't have to forget, you don't have to worry about the names. of the flies that are, we can change it. The idea of this is that you need to know exactly uh, anything about this. The only thing that you need to know, like you need to know anything about flies, what exactly is this mutation. The only thing that you need to know is that here, you see this plus, this means that is in this notation means that this is wild type. Yeah, so this is the wild type, this is the wild type, and we're working with two traits, eye color and bristles. So the wild type will be red eyes, and here we have a scarlet that is a little bit more orange eyes. Yeah, and here we have bristles that are wild type, and the bristles that are wild type are they have a combination between short bristles and very long ones. But in the mutation spineless, if you see, these bristles are much smaller. Yeah, the only thing that you need to remember is this, that they give you that information. So here you can see that here you have what? Here you will have the wild type genes. Here you have, this one will be the mutation. And here you have mutants. And here you have a mutant. Okay, that is important. And I will show you what in a second, why this is important. And as I said to you before, the names, or you need to know anything about the mutation, or yeah, look, there are hundreds. This is just few examples of mutations that are in Drosophila. This is for the eye color, and these are for the bristles, and there are many, many more. Okay, so this is just a trait that is traits that are using in this exercise, so don't worry about knowing every single detail. Okay, so now the key information here, as I said before, is that here you have ones that are normal, yeah, and here ones that are double mutant, yeah, and these one are females. Good. So one important component of the cross is that. The other important component of the cross is the males, and the males are double mutants here, and here they're also double mutant. Okay. In other words, to simplify this more, the females are heterozygous. Yeah, one have a wild type, and the other ones are double mutants. While the males are homozygous. Yeah, both are double mutants. You find double mutants in both chromosomes. Okay, so that is important. And why this is important? If you watch the video lecture, this will make a lot of sense. It's not go and watch the video lecture or read the chapter of the book. So imagine in a case that there is no recombination, you will have 100% of them looking homozygous double mutants and heterozygous for wild type and for these two mutants, so it will look exactly like this. And also, as there is no recombinance, you will not find this one, so you will have 100% looking like this. On the other hand, as we have recombination, now the parentals, they will still look like this, but now they will be lower than 100%. And you will have recombinants, so they will be single mutants. So one of them will be scarlet, and one of them will be a single mutant that it will be spineless instead of having the two mutants into one. And they will be higher than 0 degrees. So parentals will look the same. Now you have recombinants, that there will be a mix of this, and this is how I represent this with a green and blue and green. There will be a change. Now we have to find this number, and that finding the number, how low is this, or how high are the recombinant, is very easy. And they already gave us the information. Actually, they make our work much easier. That although the question looked difficult, the answer is very simple. Okay, so now I'm going to explain this very quickly, But I want to remind you, if you haven't watched the video lecture, you should stop watching this video and watch the video lecture to try to understand this in more detail. Here, to understand this question, you need to know that here are the parents. Yeah, these are the parents. And if you see here, you see that they cross a female that is white eyes and a male that is a mutant that is called diminutive, that a male diminutive that make uh, the flies much smaller. And look here, you have the results are you have parents, and the parental types are here again: white eyed with normal size, and here normal eyes and diminutive size. Yeah, so here are the same type of experiments, the same parental, you're going to get them. However, they're also going to get recombinants, and the recombinants in this case is going to be a mix different from the parentals. In this case, for example, you're going to get the flies that are completely wild type. These are completely wild type for both, and this is a double mutant in this case. Why? Because they are different from the parentals. Okay, so this is slightly different to the one that we are doing, because in the one that we are doing, we start with double mutants. Here we start with single mutants. Okay, but the idea is that you have parentals, and those parentals are not going to be 100%. In this case, they're very close to 100%, and the recombinants are going to appear and are going to be lower than are going to be higher than 0, because but in this case, how they calculate, so if you see after making all these crosses, later if you watch the video I explain why they make this cross, but the project is here. So you get a lot of parentals and very few flies that are recombinants, and in this case they count 1,000 flies. Okay, so now the key here is to calculate the frequencies, and how to calculate the frequencies, super easy. So here you, for example, take at the two parental ones that you see here, 497 plus 472. When you add them, it's 969, and you divide it in the total, so you get a percentage of 90.6 or 97. And here you do the same thing: 12 plus 19, so it's 31, and divided by 1,000, so you get this 31 divided by 1,000, so you get a recombination type of 3.1. And if you watch the video lecture, you will see that all these measurements are very similar. Yeah, all of these ones. And here in the case, this is the one that they use for flies, for centimorgans, that are the ones that use Morgan. So here is the distance, if you go here, between the white eye of the flies and the other gene here, the DM here, and it's 3.1. So the idea is that if they give you already that there is a 3.1 recombination between these two genes, they already did most of the job. Yeah. So let me show you that. Although I explained this very fast, the question you might already have it. So in our case, they didn't give us any data. Yeah. Imagine they gave you that here is 14 centimorgans. So here, this one will be 86, and this one will be 14. And why? Because these need to add 100%, and they already told you that the two genes that we are studying, that are Scarlet and ST, the distance is 14 centimorgans. So the recombinant will be, this will indicate the distance. Yeah. and the rest will be the parentals. So if you just subtract 100 and minus 14, let me draw it better. 100 minus 14, this will be 86. Good, so pretty easy. Okay, now in conclusion, you have we already have the parentals, and we had a recombinance, and we already, this was what we were expecting. These are each one of those, and now we know also the not only the genotypes, but we know the proportion. So here the parentals was the proportion was 86% of the flies. So if you divide it into 2, you're going to get 43 here and 43 here. If you are studying the recombinant, this will be 14%. So 7% of a single mutant, and 7 that it will be scarlet, and 7% of the spineless. And remember, these data points, if we study them in a lot of detail with many fruit flies, maybe this will be 40.3.56, that this can be, you know, 44. So in the question, so these are relatively not relative values, but there is some, there could be some variation, because the percentage of recombination depend on how many flies do you work with. But so this is why in the answers, sometimes you can find answer that here I gave you 43, but in the answer it could be 44. Yeah, it's very close, and it could be depending on how many flies did you study. But that was the correct answer. And the MOOC is just trying you, when they give that option, is that you know that you know the exercise very well, and you know this can have some variation. Okay, so I think that is, I want to explain it very slow, but if you have any question, let me know, and we can meet and answer some this or other questions that you have. Okay, hope you have a good day. Bye.